Now I would like to call Dr. Wilson, Brian Wilson. It's a pleasure to receive here our Biophotonic School. He's a professor at Department of Medical Biophysics from University of Toronto. His research has focused on photodynamic therapy, fluorescence endoscopy, novel photosensitizers, nano-engineered imaging probes, optical diagnostics and imaging, optical microscopy. Thank you, Professor.
Thank you. Thank you very much, and it's very nice to be back here. This is actually I think the fourth time that I have visited the university here, so it's always a pleasure to come back. This is going to be a very different talk from the one that Tim gave, which was very detailed. This time I'm going to step back and talk about a subject of nanoBiophotonics in a more broader sense. I will talk a little about PDT right at the end. How do I make this work? This should advance. Maybe I'll just use the arrows. That's okay. I'll just use it. Okay. Okay. Thank you. So there we go. So nanoBiophotonics is the intersection between photonics, nanotechnology, and life sciences and medicine. That's clear. And I just wanted to make the point that if we look at the different areas of biophotonics, of optical therapeutics, diagnostics, analytics, and biotechnology, these biomedical applications are very strongly driven by the technology that comes from photonics. So from the basic science and technology of optics. So it's really when photonics is combined with the other areas of science, whether it be bioengineering or molecular biology, that you start to be able to do things in biophotonics. And so what I want to focus on in this talk is the convergence of nanotechnology with photonics in order to impact on these different areas of therapeutics, diagnostics, and analytics. So first of all, we'll start with nanotechnology. What is nanotechnology? And then the rest of my talk, I will talk about nanoparticle-based optical analytics, diagnostics and therapeutics. And then I'll take three different areas to illustrate these advances. These are largely from our own research and development, but they also draw on other people. And then finally, if we have time, I'll talk about nanoparticles in relation to PDT. So nanotechnology, Richard Feynman, who was a very famous theoretical physicist, wrote way back in the 1950s about the potential of nanotechnology. He was really the first person to recognize that if we work in the very small scale, there's lots of things to find out that we didn't know. And he thought about that and discussed that in the context of physics, but it's also clear that there's plenty of room at the bottom. There's plenty of opportunity for nanotechnology also in biology and medicine. And in fact, nanotechnology in the form of nanoparticles were first used in the clinic in 2005. So nanotechnologies are already used in a clinical setting. And I'll give you some examples later. About 10 or so years ago, as a result of the advances that material scientists particularly made in developing nanotechnologies, National Cancer Institute in the US actually allocated 5% of its budget, which was a huge amount of money, to nanotechnology applied to cancer. And that really stimulated a lot of work. In order to define the field, NIH said that nano meant between 1 and 100 nanometers. So if you were at 101 nanometer, you could not get funding from NIH for nanotechnology. So this was just to try to restrict the field. So what does that mean? So here is 1 to 100 nanometers. This is just a size scale, a logarithmic size scale going from a tennis ball down to single molecule of water. And in the nanoscale, we're really talking about structures that are between the size of a modest molecule and about the size of a virus. 2 or 3 orders of magnitude smaller than a cell. And so you have to, when you think about nanotechnology and particularly nanoparticles, you have to think these are extremely small objects, even on the scale of a cell. Nanostructures are known in the natural world. Examples here, if you have a gecko, a little lizard walking up the wall in your bedroom tonight, it is using nanotechnology because on its feet it has nanostructures which allow it to grip on even glass. Bacteriophages, which are viral-like objects that invade bacteria, are little nanomachines. They're little nanorobots. And if you look at the color that you see in the insect world, for example, the color of a butterfly wing or the color of an iridescent beetle, that is nanotechnology. Because the color of a butterfly wing does not come from a dye within the butterfly wing. It comes from the nanostructure of the wing. And that's why if you look at a butterfly wing, it looks different color if you look in different angles. If it was a dye, a blue dye in there, that would not happen. And so that is, these are examples of the use of the natural world of nanotechnologies. If you look at synthesized nano, in other words, man-made, artificial nano, there are really 2 different categories. Excuse me. There are nanomachines and nanopattern devices. And this is an example of a nanopattern device. For example, these are cells growing on a structure that is patterned, nanopatterned. And it has been shown, for example, that if you grow cells on a structure, on a surface that is nanopatterned, the molecular biology is completely different from when the same cells are grown on flat plastic. Big question, which is correct? Is the nanostructure molecular biology correct, or is the flat plastic molecular biology correct? But one of them is not the same as happens in the living object. If you look at nanomachines, of course, this is what we think of as the real nano world. Isaac Asimov wrote the book Fantastic Voyage, and I'm sure many people have seen the movie of Fantastic Voyage of injecting nano people into the bloodstream. Is this scientific reality? If you look at Google, and I did this last week, if you type in nanobots, which is a synthesized word meaning nano-sized robot, if you type in nanobots in Google, you get 1.8 million hits. So you think, wow, that's a big subject. If you type in nanobots in PubMed, you get 10 hits. Right? So this concept has really not yet made it even into biomedical research, but it's starting to. So I just cite this paper, for example, as looking at self-powered nanostructures that is respectable science, and it is starting to impact on biology. The other area of nano, man-made nano, is nanoparticles, and I'll just give a number of examples here. I'll talk about this in much more detail, but just to make the point that these are nanostructures, nanoparticles, on the 1 to 100 nanometer scale, which are very different. So there's lots and lots of different types of nanoparticles, and in fact, it's this that I want to concentrate on today. Important to recognize that nanoparticles are already used in the biological world and in the clinic. Many sunscreens have nanoparticles in them. There is at least one pregnancy test kit on the market which is based on nanoparticles to try to modify the sensitivity. And in fact, a number of drugs have reached the market that are based on nanotechnology, nanoparticles. And that includes PDT drugs, and I'll just illustrate that a little later. So, if we look at nanoparticles for biology and medicine, what do? There are 2 basic functions. One is they can serve as active agents. They do something by themselves. And the other is that they deliver, like a pizza delivery. If you look at the active agents, this is based on the fact that if you take a given material, then one or more of the physical properties of that material may be very different from bulk material. This is illustrated by this very famous example. This is a glass cup called the Lazarus Cup. It's in the British Museum. And it was made by the Etruscans several thousand years ago. And the interesting thing here is if you look at this cup in transmitted light, so if you hold it up, you're not allowed to because it's in a big safe, but if you hold it up, it looks red. But if you put a light inside it, it looks yellow. Or reflected light looks yellow. And that's based on the fact that this glass contains gold nanoparticles. So the ancient Etruscans knew empirically that the optical properties of gold were different if they used it in a colloidal form, which is now we understand as nanoparticles. So the physical properties may be very different from bulk properties. This is usually manifest in so-called hard nanoparticles, either metals or semiconductors, ceramics, carbon, and so on. How is this used? Well, here's an example in radiotherapy. So this is a study published a number of years ago in which gold nanoparticles were used in order to enhance the radiation dose delivered to tumor. And so in these mice, gold nanoparticles were injected. This is a tumor here, and then they were irradiated. And this is the growth curves. If you have nothing done, radiation treatment only, gold plus radiation, and the corresponding survival curve. So this is the survival curves as a function of a high dose of gold, a lower dose of gold, and then no gold. So there's no doubt that if there is gold nanoparticles in the tumor, radiation is more effective. And in fact, gold nanoparticles are an amazing radiation sensitizer. The physics of this is still not quite understood, but there's some understanding. Nanoparticles are used in MRI, magnetic resonance imaging, as contrast agents. These are paramagnetic gold nanoparticles, and they change the relaxation properties of the tissue, particularly water, and can be used therefore as contrast agents. That's a standard material now in NMR. If we look at the other aspect of nanoparticles as delivery vehicles, this is really the guided missile of medicine. So this is like the North Korean version of nanotechnology, where they have a certain payload. In the case of North Koreans, it's a nuclear warhead, and they want to deliver it in a targeted way. They want to deliver it to South Korea or to the US bases. Okay, so this is the idea of targeted delivery. And this is just the general idea that you take nanoparticles, you load them up with whatever you want to deliver, whether it be drugs or genes or contrast agents, and you target them using some sort of agent like an antibody that's on the surface, so that when they're administered, they will target to, for example, the tumor. And you also have other coating on the nanoparticles, for example, so that they are not just taken up by the liver and excreted before they can get to the target. Most of these delivery vehicles, or not all of them, are soft nanoparticles, nanoparticles made from lipids or polymers or lipoproteins, but there are also hard nanoparticles, such as gold, for delivery of drugs. Examples of this, in fact, there's a nice review article on this, lots of different nanoparticles, liposomes, lipoproteins, nanoemulsions, etc., polymers have been used to deliver drugs, and each of them has different properties. Just one example, here's a dendrimer, which is this complex spider web-like structure where the molecule of interest, the drug, for example, is incorporated into this nanoparticle, and this just shows the growth curves of tumors. This is without nanoparticle delivery, essentially this drug, which is methotrexate, which is a standard chemotherapeutic drug. Millions of patients receive this drug. In fact, in this particular animal model, you get no control over this particular tumor, even at doses of the drug that are lethal. Whereas if you deliver the same drug in this dendrimer structure, you get significant tumor response and non-lethal doses. This is another more complex example where gold nanoparticles were, first of all, decorated with a polyethylene glycol so that they would have a long circulation time, and then were used to deliver tumor necrosis factor, which is an active biological agent. And this just shows the nanoparticles. You see they're colored because this is gold, and different size nanoparticles have different color. And so you see, you can actually see the gold nanoparticles from the coloration of the tumor. So there's no doubt that the gold nanoparticles are delivering the drug to the tumor. So what about now optical? So let me concentrate from the rest of this talk on just the optical nanoparticles. So optical nanoparticles are used in non-biological applications also. This is an example of using gold nanoparticles on photonic crystal fibers. So you essentially take optical fibers and coat them with nanoparticles. And why one would do that is the fact that the nanoparticles have very specific spectral characteristics, very particular optical characteristics, and you can change the wavelength transmission characteristics of that optical fiber in a tailored way, however you would like to do that, by coating it with nanoparticles of different sizes. These are examples of optically active nanoparticles, in fact, metal nanoparticles. And the metal nanoparticles, as you saw from the example of the Lazarus Cup, the metal can essentially, depending on the size and the shape of the nanoparticles, can have different colors. So we think of gold, for example, as gold-colored. Right? It's a sort of yellow color. But in fact, if you take nano gold, it can be any color. So you could make blue gold, yellow gold, red gold, near-infrared gold, whatever you like, just depending on the nanoparticle size and the shape. And of course, it also depends on the material. And this is just illustrated. So these are nano rods, for example. They're all rods of a metal. And you can see, depending on the size and, in this case, the aspect ratio, you can make it any color you like. So you can vary the optical properties, and this is just shown by the spectra. Another example of optical nanoparticles are quantum dots, which are made of semiconductors rather than metals. And in this case, the quantum dots are actually, they're usually a core and then a shell around them. And the point about these is that they're very, very highly fluorescent. And I'll come back in a little while and show you how we can use that in biological applications. They have very relatively narrow spectral emission, fluorescence emission, and they have a broad absorption band, and it turns out that's quite useful. Coming now to the main topic of biophotonic nanoparticles. Now, we're just talking about optical aspects, and we're just going to concentrate on the bio. So these are photonic nanoparticles that are either bio-targeted or have biological activity. Here's an example of a drug delivery by nanoparticles. This is actually from our own lab, where we made lipoprotein nanoparticles. And you can target those, for example, by antibodies or by peptides, whatever. And this just illustrates that these genuinely do have targeting capability. So this mouse had two tumors. In one of the tumors was missing the particular antibody receptor, and the other one expressed that antibody receptor. The nanoparticles were injected. These are fluorescent nanoparticles, and you see that the nanoparticles only go to the intended target, only go to the tumor where the antibody is actually expressed. So there is true targeting by this technique. Now, of course, you could say, well, I'll just take the antibody and the fluorescent dye, and we'll get equivalent targeting. But the point here is that you can load up this nanoparticle with, for example, a drug, or with a virus, or with a gene, to do a therapeutic effect at the same time. Another example of using optically active bio nanoparticles, these are quantum dots. used for lab-on-a-chip diagnostics. So here you might be interested in an ex vivo diagnosis using a sample of urine or a sample of blood plasma or even single cells. You have very little material to work with. You want to fluorescently label that material to give you the information, and you want lots of information. And so you want to be able to so-called multiplex. And so this is just like barcoding. You use the fact that these quantum dots of different sizes, each of a different color, and you have each quantum dot, for example, linked to a different antibody, so that when you then put that in as a probe in a lab on a chip, you read out a lot of independent information, which tells you a lot about the molecular expression of the cell or the sample. So let me take some examples from our own R&D, and I'll just continue with the quantum dot story for the so-called multiplex bioimaging. So what are the advantages of quantum dots for fluorescence bioanalytics and imaging? So first of all, they're very bright compared with fluorescent dyes. They're very highly stable. They have low photobleaching. This is illustrated here. This is a cell in which a dye, an organic dye, a fluorescent dye, Alexa, has been used to stain structures in the cytoplasm, and quantum dots have been used to stain the nucleus. And as a function of light exposure, you see, in fact, that the dye just bleaches away, whereas the quantum dots actually maintain their brightness. Sometimes they can even get brighter with light exposure. So this is very nice if you want to do long-term tracking, long-term following of cellular processes. You can use quantum dots for that purpose. They have relatively narrow emission spectra. As I said, this allows you to do multiplexing. And they have broad absorption spectra, so it means it's economic, because you could, for example, use a single laser source in order to excite all these different nanoparticles. You don't need a different laser for each dye. Limitations are potential toxicity. Quantum dots are made of semiconductors. They have fairly nasty rare earth materials in them, and so there is concern about toxicity, which is likely to limit the clinical use, although it's not impossible that quantum dots could someday be used clinically. They are relatively expensive, and because it's a semiconductor, it's actually not so easy to link biological molecules onto a semiconductor. So there's a bit of trickiness in terms of the surface chemistry. But nevertheless, they're useful, and so this idea of quantum dot multiplexing is around, where basically you take 3 different colors of quantum dots, different sizes, you label each one of them with a different antibody, you shake them up, just like in a bar, you get a cocktail, and then you administer it, and now the different antibodies will target different aspects of the disease that you're interested in, and so from the color, you're able to say, okay, this area of the tissue expresses this antibody. This area of the tissue expresses this antigen. This has been demonstrated a number of years ago by Shuming Ni initially, looking at different colors of quantum dots. This was a kind of artificial experiment. This is not a real targeting, because he really just injected these different antibodies into different sites. But here's an example from our lab of using different quantum dots, 3 different colors of quantum dots in colorectal cancer, each with a different antibody. And you see that there is non-uniform expression of the antigens throughout that colon tumor. So if, for example, you're going to use a drug which targets and interacts with one of these receptor molecules, it's not going to be very successful. It's only going to target part of the tumor. And that's a big, big problem of tumor heterogeneity and the use of drugs. This is a study which we did to see whether quantum dots will be useful for multiplex histopathology. Fluorescence histology is very seldom used in clinical practice, although it's starting to be. And one of the problems has always been that the pathologist says, I don't like to use fluorescence because I can never rely on seeing the signal properly. So it photobleaches or it's not bright enough, etc. And so the idea here is to use quantum dots, and again, just as in the mouse model, you use multiple quantum dots, a cocktail of quantum dots with different expression biomarkers, and you see the sort of very beautiful results you can get. This is a tissue microarray where this is a blown up image of just one of these spots, and you can actually correlate the actual messenger RNA level in these cells, in these tissues, with the quantum dot fluorescence. So this sort of relationship tells you for the first time, perhaps I can do quantitative histopathology. So as opposed to, instead of pathology being a qualitative, subjective science, you can for the first time start to say, could we make pathology an objective quantitative science? This is an example of the fact that quantum dots photobleach allowed us, and I don't have the video here unfortunately, but this is a long-term video looking at the interaction of natural killer cells, which are part of the human immune system, with the leukemia target cells. And if we had to move it, you could actually see the natural killer cell enveloping the tumor cell and digesting it. And you're able to do that with quantum dots because of the fact that they don't bleach. And so you can monitor and expose the cells to light in the microscope for a long time. And this was some work that we did to look at quantum dots in vivo, for example, in colon tumor detection in a mouse model, and in brain tumors as a contrast agent to guide brain tumor surgery. We haven't moved quantum dots to the clinic yet, but we're certainly using fluorescence brain tumor surgery in the clinic, and I'll talk about that on Friday. So, back to the outline. So the second example is one that we're very enthusiastic about at the present time. We think it's a really interesting new possibility, and that's to use so-called SIRS nanoparticles in lung cancer. If you're not interested in lung cancer, but you're interested in cervical cancer, you can apply the same concept. Okay, we just happen to have started this in lung cancer. So SIRS stands for Surface Enhanced Raman Scattering. So to understand SIRS, one has to first understand Raman Spectroscopy. Excuse me. And the Raman Effect was first described by Raman, who was an Indian physicist and Nobel laureate. He won the Nobel Prize in Physics in 1930. And what Raman discovered was that when light interacts with a medium, such as this laser light hitting my hand, what you see is mostly light that is elastically scattered. How do you know that? Because this color is the same as this color. So the light that you're seeing from my hand has been multiply scattered. If I put a mirror up here and risk blinding you all in the audience, you would be blinded, you would think the color is the same. So the light that's multiply scattered and comes out here is elastically scattered because it has the same color. However, about 1 in 1,000 of the photons is actually fluorescent. So if you were to put in front of your eyes a filter which removed the green light, you would see a little spot of red light on my hand, and that would be the fluorescent. And that's about 1 in 1,000. About 1 in 1,000,000 of the photons are inelastically scattered. Okay, and that's what Raman first described. And inelastically scattering is actually the interaction of the light photons with the vibrational and rotational energy levels of the molecules. So when you look at fluorescence, what you do is that the photon is absorbed and excites the molecule to an electronic excited state. Sometime later, the molecule de-excites. So that's a two-step process where the photon is first absorbed and then a second photon is emitted of longer wavelength. In the case of Raman, the same photon simply is slightly wavelength shifted. These wavelength shifts are very small, but they're very important. And they're important because if you look at our typical Raman spectra, this is what you see. So this is a Raman spectrum from a single organic molecule. And what you see is this rich information. If we compare that with the fluorescence signal from a quantum dot, it has very little information, but also it's extremely broad. Whereas the Raman spectra have a lot of molecular information, and the lines are very, very narrow. And that is a big advantage. So a number of people, including ourselves, in the 90s and early 2000 were interested in using Raman spectroscopy as a means of early cancer diagnosis. And so this is an example of a system, fiber optic based system, that was built to go into an endoscope, and you see it here, this fiber optic probe coming down and touching onto the tissue, in this case inside the esophagus, and it's making a Raman measurement. So it's putting light of one wavelength down, and it's looking at the slightly wavelength shifted light that comes back up the optical fiber. You have to, from an engineering point of view, this is a little tricky because the signal is so small, it's only 1 in a million, that you have to be worried about lots of different background factors. But if you do it properly, you can extract these Raman spectra. And these are Raman spectra measured in patients. And you can see that there are differences. In fact, there are disease-specific differences. So in the case of, for example, in the esophagus, the condition that you heard about this morning of Barrett's, where PDT is used, where you're interested in detecting these very small premalignant lesions. In fact, Raman spectroscopy has greater than 90% specificity. And so you think, wow, that's marvelous, that's solved a clinical problem. And it has solved a clinical problem, except it's not really clinically practical because the signal is so weak that it takes about 5 seconds per point. And so that's problematic just for clinical practice. And you cannot make an image in that way. So there's a very beautiful paper from England a number of years ago where they took samples of the tissues from these patients and did a microscopy, Raman microscopy of the tissues and demonstrated, confirmed the fact that these, you can make Raman images that demonstrate the disease, but the imaging took 4 hours. To do just a little biopsy specimen. So that it's conceptually, it's a great idea, but it's not practical. Is there some way of making it practical? And this is where nanotechnology comes in. So it turns out that if you take a metal nanoparticle and you put on the surface a molecule that has a Raman signature, and any organic molecule has a Raman spectrum, then the signal, the Raman signal, is enhanced by a very large amount. Now, I put here greater than 10 to the 4 enhancement. There are reports that the enhancement can be as high as 10 to the 15, depending on the exact conditions. The enhancement factor depends on a lot of different things, including the shape of the nanoparticles, and essentially comes about because as the light, which is an electromagnetic wave, goes past these metal nanoparticles. Metals have free electrons, the free valence electrons, and so in fact the electromagnetic wave in fact makes the electrons in the nanoparticles oscillate, and if the wavelength of the light is correctly balanced against the size of the nanoparticles, you can get a resonant behavior. It's a bit like pushing a kid on a swing. If you push the kid each time they come back, then you get the swing gets bigger and bigger and bigger. So you're amplifying the signal. And so the same idea. And so we take metal nanoparticles, such as gold nanoparticles, and coat them with a Raman molecule, and then look at the signal. Now, the definitive paper on this was published in 2008 in Nature Medicine, I think it was, where a group took a couple of mice with tumors, and in one mouse they injected quantum dots. And remember, I told you before that quantum dots are extremely bright fluorescence. And then in the other mouse they injected the surface-enhanced Raman nanoparticles, and the optical signal in the nanoparticle case was 200 times stronger than the optical signal from quantum dots. So that was a completely remarkable finding that no one would have expected. And actually, previously we thought of Raman as a very weak process compared with fluorescence. Now we think of SERS as a very strong process compared with fluorescence. So it completely changes your thinking about using fluorescence versus using SERS. So the particular clinical example that we have been working on is in lung cancer. So we like to do things that are driven by a particular unmet clinical needs. So in lung cancer, as everyone knows, it's a terrible problem. This 5-year survival is dreadful. It's much better if you can detect it early, but the problem is the detection of early lung cancer. Endoscopy is used to try to detect early lung cancer in the central airways. But in most cases now, in the peripheral airways, early cancer is detected by CT scanning, so X-ray CT screening. And so a number of countries have screening programs where high-risk patients, for instance, heavy smokers, get CT scans. So whole chest CT scans. And this is an example. So you see this little area of contrast here, and there's another area of contrast here. And you say, wow, that's fantastic. You know, the CT scanning really works. But the problem is that, in fact, over 90% of these are false positive. They're not tumor. And so the patient is faced with the dilemma, okay, I have something in my lung, so I have two choices. Either have a big needle put through my chest wall so that it can be sampled. That's a fairly dangerous procedure. Or I wait. And I have it done next year, and if it grows, maybe it's cancer, maybe I should have it. So that's not a good choice. So the idea is, could we use SERS bronchoscopy and go way down there and tell whether or not that's a tumor? The challenge here, and the reason why we're using SERS, is the following. That if you look at molecular targets in lung cancer, so we all know about biomarkers and molecular targets, if you take 100 people with lung cancer, you say, well, you've all got lung cancer. But if you take the tumors and you do genetic profiling, and you look at the molecular characteristics, you find, in fact, that no two tumors, no two patients have the same molecular profile. And this is just illustrated here. This is a pie chart showing the fraction of patients which express different biomarkers. And it's been shown that if you want to have a detection test that has acceptable clinical accuracy, then you need at least 3 biomarkers, and preferably 5 or even 10. So you need multiplexing. And that's where the Raman comes in. Because of those very, very narrow lines, you can think about multiplexing. So the concept is gold nanoparticles with Raman reporter molecule, and a coating, which essentially is to protect them, targeted by some sort of antibody or peptide or whatever. And just as in the case of the quantum dots, you can make different nanoparticles with different reporters. And this shows, for example, the overlapping spectra of 3 different Raman molecules. And you shake them up, and you would deliver those as a cocktail. And now, because these lines are so narrow, you can actually separate them. And so you can pull out the information. This sounds easy, but in fact it's turned out to be extremely difficult. So we've been working on this for 5 years now, because the challenges are in materials science, in biochemistry, in molecular biology, in instrumentation, in scale-up, blah, blah, blah. So even the animal models, we had to reinvent some of the animal models. But nevertheless, I think we're probably about maybe 2 years from the first clinical study with this. But in the preclinical setting, you should ask a number of questions. Does this work? Could you actually target SERS nanoparticles? And the answer is yes. This shows it in cells. So these are tumor cells, and you see the spectra in the cancer cells. And this is in lung tissue. So here we simply took a piece of human lung tumor and dipped it into the nanoparticle solution. And this is untargeted, and this is targeted. And you see that there is actual uptake in the lung tumor. We use this mouse model to look at the targeting. So again, as in the case I showed you earlier, this is just two different fluorescent proteins. This is a non-targeted tumor, and this is a targeted, and you in fact get targeting only to the tumor that expresses the particular antigen. There's a lot of work being done in the instrumentation side. I won't go into details on this. It's recently been published, but we've shown that you can do at least 4-plexing. In other words, four different nanoparticles with about 30 picomole sensitivity. And we think that that's high enough sensitivity to actually be able to pick up the biomarkers, because you have to think, well, if the nanoparticles are going to be delivered to the tumor cells, there have to be enough receptors on the tumor cells to give us a decent signal. And it looks as if that probably is the case. And we're now working on a new endoscope system. Oops, can I go back? How do I go back? We're working on a new endoscope system in collaboration with Eric Siebel at University of Washington, which is a completely new idea for endoscopy. Rather than a fiber bundle or putting a video chip at the end, it's actually a flying spot endoscope. And the advantages of this are that you can essentially measure any optical signal, so we can measure the Raman signal, for example. And the other advantage is you can make these extremely small. The one we have at the moment is less than 2 millimeters diameter, so we think we can really get deeply down into the lung. So if I come back in 2 years' time, maybe I'll show you the first patients. So the third example I'll give is multimodal nanoparticles for cancer diagnostics. And this was work that was first published in this article in Nature Materials in 2011. And these are nanoparticles that are made by taking a porphyrin, and we heard all about porphyrins for PDT, so it's the same material. In fact, the one we're using at the moment is almost the same as the TrueCAD that's being used for prostate cancer. And you link that to a lipid, a fat. And you shake it up, and nanoparticles are formed. That's not too surprising, but there was something that was very surprising, is that you can get 100% packing. So every component in this nanoparticle is made from these porphyrin lipids. So there are 80,000 porphyrin molecules in every nanoparticle. That is a huge number of porphyrin molecules. The reason is that the porphyrin molecules are flat, and so they can stack together inside the shell. I put this slide up to illustrate this guy, John Lovell, who is a PhD student in our lab, who discovered this. And the reason I put this up is to impress upon the professors and the students here that it's very important that the students are allowed to play in the lab. Playing in the lab is good. So if your professor says, no, you cannot play, I want you to do this, you tell them, we'll never discover anything that way. You have to let me play. And so John Lovell was playing in the lab, and he just asked the question, how many porphyrin molecules can I pack in before the liposome falls apart? And the literature told you 15%. You can't get more than 15% of any molecule into that shell before the particles will fall apart. It's not true. You can get 100% in, and as a result of that, in fact, John got a tenure-track faculty position before he had even finished his PhD because of playing in the lab. So please play in the lab. So there's lots of different applications of this nanoparticle, and the reason is it can do many, many different things. And in fact, the editorial in Nature Methods said this is one particle to rule them all, the same as The Lord of the Rings. The very high porphyrin concentration means you can do photoacoustic imaging. I'll tell you about that in a moment. You can do enhanced photothermal therapy. You can do photodynamic therapy. You can do fluorescence. You can load the porphyrins with copper-64 to do PET imaging. You can load them with manganese or gadolinium to do MRI, and you can target them. So in a single nanoparticle, you can do many, many, many things. And that's great because that means you can get all those functions happening in a single agent. This just shows you examples. This is photoacoustic imaging, which is the use of pulsed laser light, which then generates, because of microheating, generates ultrasound, which you can image. And this just shows porphosomes, even without targeting, are taken up in lymph nodes. And this shows photoacoustic imaging of porphosome in both HER2 positive breast tumor, that is breast tumor that expresses a particular very important antigen, and HER2. And you see that, in fact, the porphosomes go to the HER2 positive tumor. They can be used to enhance photothermal therapy, which is in a way a competitor for PDT. Because they have such very, very high optical cross-section, they will absorb near-infrared light. This shows the temperature in the tumor. If you have a laser, a near-infrared laser, the temperature rises only a few degrees. Without the porphosome, it goes above the coagulation threshold. With porphosomes, and you can then cure the tumors, at least in mice. So that's become a big area of work for us as well. So let me finish with nanoparticles and PDT. And actually, there's a nice article written by Professor Bagnato and Dr. Allison that I refer to you, 2008 article on photodiagnosis and photodynamic therapy, 2008, although 5 years is a long time in nanotechnology, so Vandali, you have to update this paper. Right? But it's a very nice starting point and has lots of references. So, have nanoparticles been used in PDT? The answer is yes. And the question is, what can you use them for in PDT? Well, the first is obvious. You can use them to target the photosensitizer, just like any drug. Plus, with or without another imaging or cytotoxic agent. And I'm going to give you examples of these. Secondly, you can use it as a kind of optical energy transducer or an antenna. Take light energy and transfer it into useful work for you in terms of the biology. You can actually use nanoparticles as the light source, which is quite interesting and a little unexpected. And you can actually use nanoparticles as the photosensitizer itself. So let me just illustrate those four possibilities. So photosensitizer delivery, just as with lots of other drugs, photosensitizers can be delivered by nanoparticles, and in fact, two of the PDT drugs that are in the clinic now, Visudyne for macular degeneration, and Foscan, which is used for many things, but includes, for example, nasopharyngeal cancer treatments, head and neck treatments, are actually nanoparticle based. Because these molecules are actually not very water soluble, it's hard to deliver them, and so they're packaged in a liposome in order to make them biocompatible, and so that they can distribute to the body, go to the tumor or the target cells, and then the drug goes into the cell via endocytosis. So the membrane, the lipid membrane, and the lipid membrane of the cells merge like two bubbles in the bath, combining to make a larger bubble, and the drug, the sensitizer, is delivered into the cells. So those are almost standard techniques now. Here's an example from Tayyaba Hassan's lab, where they've gone one step further, and they've combined a liposomal delivery of the photosensitizer, which here I guess is these star-shaped objects in the shell, with another active agent. So they've put in a chemotherapeutic agent in here as well, so you're delivering both the sensitizer and the chemotherapeutic agent, and they get much better tumor response than either the chemotherapy or the PDT alone. Here's an example of energy transduction. So this is carbon-60 buckyballs, famous discovery by Bucky in the 60s. And so you can link buckyballs with other molecules and make them photosensitizers. This shows the absorption spectrum of this particular nanoparticle construct. This shows that they do generate singlet oxygen, and this shows that you do get light and drug dose-dependent cytotoxicity. So it is a valid approach. They have certain advantages and disadvantages, that they are relatively short wavelength, buckyball absorption, and it's difficult to push it out much, but there may be applications, as you heard this morning, where the short wavelength is actually an advantage. Another example of energy transduction in PDT is quantum dots. So it turns out if you link quantum dot to a photosensitizer drug, the quantum dot can have a very high cross-section, can absorb the light, and then give energy transfer to the photosensitizer molecule, and again you'll generate singlet oxygen and do the biological effect. The advantage here is that the optical absorption of the quantum dot has a very broad wavelength spectrum, as we saw earlier, and therefore is much less dependent on an exact matching to a particular laser source. This is one that surprised me, and in fact is very, very recent. This is a 2013 paper that I think just came out a very short time ago. This is the idea of nanoparticles as the light source. So wouldn't it be nice if we could get rid of the lasers? The companies wouldn't like it so much, but the rest of us would like it. Why not actually generate the light inside the cells? This is not an entirely new idea. Sandy McRobert in University College London, where Professor Hopper comes from, a few years ago suggested and published a paper on so-called bioluminescent resonant energy transfer. But that was non-nanoparticle based, and it'll be interesting to see whether this has an advantage. So essentially what you do here is you take quantum dots and you decorate them with the bioluminescent substrate. So I think many people have heard of bioluminescent. If you haven't, we can talk about it. But it's the same process. It's a chemical process that is used in insect signaling, in fireflies, and light is given off as a result of a chemical reaction. So the idea here was to have the quantum dots coated with the substrate for bioluminescence, and then you have the photosensitizer as well. Now what happens here then is that the light is absorbed by the quantum dot, and the energy is transferred actually to the start again. What happens here is that the bioluminescence reaction results when you give the complementary material, such as luciferin, into the body. You get the bioluminescence reaction, and that light actually is absorbed by the quantum dot and transfers the energy to the photosensitizer. If you're a very serious physicist, you may argue that this is not actually a light source. Because this is actually arguably not correctly drawn, because probably a photon is not emitted and then reabsorbed. It's just that the photonic energy that would otherwise be given off as light is in fact used and transferred directly to the photosensitizer. So it's this so-called resonant energy transfer process. Nevertheless, the process works, and in fact it's because of the fact that actually a photon is not given off and then reabsorbed that makes this possible in practice. Because if you think about it, if you think instead of having a 2-watt diode laser, we're now going to take some of the molecules that a firefly uses to glow in the dark. You think there's not going to be enough light. Right? In fact, we showed 20 years ago there's not enough light. But if the photosensitizer is very close to where that photonic energy is generated, then that process can be very efficient. And so you can amazingly produce enough energy to activate the photosensitizer and get a biological effect. So it's a very nice idea. Still quantum dot dependent, which leads to toxicity questions, and there are some questions about whether one would be happy administering luciferase substrate to patients, but nevertheless, as a concept, this is very, very appealing, and we'll see how much progress it makes. And then finally, just to finish off, here's can a nanoparticle itself be used as a photosensitizer, even without a photosensitizing molecule? And the answer is yes. This is some work that we did a number of years ago with porous silica. silicon as a photosensitizer, and it turns out that if you make silicon in porous, very porous form, it has a huge surface area. So this is like a sponge. So it's very porous, nanoparticle sponge, and it turns out the surface area is so large that even although the process of singlet oxygen generation from this silicon surface is very inefficient, the surface area is so large that you produce a measurable amount of singlet oxygen. So again, this has not reached, certainly not reached the clinic stage. In fact, not much has been done even preclinically on this, but it is an example of how you could get away from the idea of having to make a photosensitizer molecule and just use nanoparticles as the photosensitizer. So with that, I'd like to also just point you to this nice publication very recently, shedding light on nanomedicine, which is a nice article on nanoBiophotonics. So I hope that's given you some idea of this field. It's a very rapidly expanding field. There's a lot of crazy ideas, and that's good. Crazy ideas are good. We'll see how many of them reach the clinic, but hopefully some of them will. So thank you very much for your attention.
Hi, Professor. My question is about the porphysomes. Is the bleaching of the photosensitizer diminished by its, because of its organization into nanoparticle?
So that's a very good question. When the porphyrins are in the nanoparticle, they're non-fluorescent. It's completely quenched. Okay. In fact, they're not photoactive. They have no PDT effect. But when they go into cells, the porphyrin structure, the liposome structure disintegrates, and the porphyrin molecules are released, and then they become fluorescent and photoactive. So the porphysomes actually tell you when they have reached the target, because they become fluorescent only when they go into the target cells. But otherwise, the quenching is very, it's a huge quenching factor. And so they don't, so equally they do not photobleach, because there's no energy transfer in that sense. Essentially all of the energy that's absorbed by the porphysomes is converted to heat, which is why they're such an efficient photoacoustic and photothermal contrast agent.
Okay. And just another question. I've seen some data on the literature concerning about the caping of gold nanoparticles, metallic nanoparticles in general, with photosensitizers. What do you think about that? For PDT and hyperthermal therapy, both. What do you think about that?
So putting a photosensitizer on the gold nanoparticle?
Yes.
Yeah, sure. The question is really, I think the comparison you have to make is whether it's better to do that, because you still have to target. So you want to then have the gold nanoparticle with a photosensitizer attached or coating, and then with, say, an antibody. Does that give you more drug in the target than simply taking the photosensitizer and linking it directly to the antibody? You can argue it both ways. You just have to do the experiments to find out. I mean, the advantage of the nanoparticle, in principle, is you can put on a very large number of molecules onto nanoparticle, and you can put multiple antibodies. But on the other hand, the nanoparticle has to be delivered to the target. So it's a tricky balance between the biodistribution and the targeting. But certainly, people are looking at that as a possibility.
But the junction of between PDT and hyperthermal therapy, is it good?
I think it depends on the application. I mean, in prostate, for example, we've done a lot of PDT work. We're currently doing photothermal, if you like hypothermia, but hypothermia above 55 degrees. We think for focal prostate cancer is actually probably better than PDT, just because it's more reliable. We think we can get more reliable results because it's less dependent on getting the exact photosensitizer dose optimized, and that varies a lot between patient and patient. If you look at treating the whole prostate to destroy it, probably PDT is better because you get the biological selectivity. So on the one hand, the photothermal is physically simpler and more controllable, but you don't have the biological specificity. So it depends very much on the particular application.
Thank you.
Hello. My question is about imaging systems. When you talk, make comparisons about dyes and quantum dots, you talk about toxicity, the photobleaching. How about lifetimes and the concentrations compared to the dyes? Are the same, are different?
You mean to use, I'm not quite sure I understand, you mean to use the fluorescence lifetime as a contrast. I actually don't know much. I don't actually know the answer to your question. Actually, maybe I don't know. John, do you know much about the fluorescence lifetime of quantum dots? How long? Okay, so 20 nanoseconds. So it would depend on the application of whether or not that's useful. I assume that the lifetime doesn't depend on the coating. Is that correct, or is that? Yeah. Okay. So I don't know, John, maybe say that again, just because people might not hear it.
So if people who didn't hear, so the fluorescence lifetime of quantum dots is generally longer than normal fluorophores. So if you think of a normal molecular fluorophore, as long as it hasn't got metals in it, it's a few nanoseconds. Quantum dots are normally longer. They're normally up at about 20 nanoseconds. But what they are very good at doing is doing FRET fluorescent-based imaging, where you put the two quantum dots close together, because then you see a really rapid change in the fluorescence lifetime, because in essence the energy is being transferred by FRET and not by the normal fluorescence process.
Another question, please. You are talking about nanoparticles and quantum dots, but there is some opinions that the sizes more than 10 nanometers tend to accumulate in body. Don't be excreted. That will be a problem or not?
Yeah, so the optimum size is a very active area of investigation with all nanoparticles. You know, we've seen, for example, with the porposomes, that the optimum is probably about 60 or 80 nanometers. If we go too much smaller, the clearance is very rapid, and so we don't get very good target uptake. But if we go too much larger, then we lose a lot of the nanoparticles in the reticuloendothelium, in the liver and the spleen. So there seems to be always a balance between the. It's really a balance between how long the nanoparticles can keep circulating and what is the avidity of the nanoparticle for the target. So I think there is some argument about whether or not you are actually better to use very small nanoparticles with very high avidity so that you would get a lot of the uptake in the target on the first or second pass, or whether you're better to use a larger nanoparticle and accept that you'll lose a lot of them in the liver, but you have much longer circulation to give time for uptake. So I don't think there's any general answer yet. It really is very specific to the nanoparticles.
Brian, I want a simple question. When we see enhancement of effects, like the Raman enhancement, is that because we have a resonance on the nanoparticles?
Yes.
And light fields are much larger?
I'm not sure what you mean by light fields are much larger.
Well, that, you know, when I have a nanoparticle system, that's like a cavity. Light gets trapped inside. Therefore, I do have some enhancement on the fields.
Um,
That's why we heat up and we do all kinds of things.
Yeah, I'm not I don't know. I'm not sure the correct way to think of it is the light being trapped. I mean, it's more that the frequency of forced oscillation of the free electrons in the metal nanoparticle.
So it's more related to the plasmonic?
Yeah, it's plasmonic for sure. Yeah. So you're forcing the electrons into an oscillation, and if that oscillation corresponds
But
to
the plasmonic field will also produce enhancement of the light field, right?
Yeah.
No?
I mean, you can get, for instance, if you take gold nanoparticles and put a fluorophore on them, you actually get an enhanced fluorescence.
No, I just want to know if the nanoparticles are actually changing the light-matter interaction on the surrounding, or it's just a question of field enhancement?
It's field enhancement.
So, okay, so it's just field enhancement.
Yeah. Although again, John, you may know better than I do. I mean, this 10 to the 50 enhancement thing that's floating around, my understanding is that there's at least 3 or 4 different phenomena that have been claimed for that enhancement.
Yes. So there's all. The truth is, I don't think anyone really understands it. There's another slight aside.
No, but people do understand in glasses, you know, doped glass. Must be equivalent.
Oh, yes, but there's several effects taking place. So the other thing is SIRS was discovered by someone called Fleischmann in 1974. He has another claim to fame, which he discovered cold fusion. And therefore all his SIRS has been forgotten to some degree. The other thing that happens, if you have a perfectly flat single atomic layer, or couple atomic layers that's perfectly flat, you don't get the same enhancement. So you do want little bumps in the surface. So you're getting local, it's acting like a lightning conductor. You get local enhancement in the electric field. And it's a combination of the two things that happen.
Yeah. And in fact, people have made, rather than making, say, a gold nanosphere,
you want rough balls.
They make gold nanostars, which have many spikes sticking out of them, and you get huge enhancement with that, although they have other disadvantages.
Yes.
But it's really, if you have a sharp structure, most of the signal enhancement is actually coming from the tip.
And that's when you get the 10, so that's when you get the 10 to the 15.
That's because of the curvature. It's still field enhancement. The plasma is more intense there, the plasma.
Yeah.
Well, the second question is more by...
That was not a simple question.
What is the time of releasing the nanoparticles in a bio object, in a cell? How long the nanoparticles stay inside? What is the clearance time?
Again, as with the other question, there is no single answer to that. I mean, it's...
because if we're going to put nanoparticle in people and they're going to live with that forever,
right.
That seems a little scary to me.
Well, that's certainly one of the problems with the quantum dots, because they have toxic heavy metals, and so even if you say, well, look, I've covered these quantum dots with glass, say, or whatever, and they stay forever, then you risk them breaking down. It's on the other hand, the metal nanoparticle, I mean, gold is used clinically. I mean, colloidal gold has been used for decades in patients for rheumatoid arthritis, and at huge doses compared with what we've been talking about here. Now, admittedly, it's not injected intravenously; it's injected locally. But nevertheless, it is generally thought that the toxicity, even if you have almost permanent capture, that the toxicity of gold nanoparticles is very, very low. So again, this gets into these complicated questions. For instance, the SIRS effect is much higher if you use silver instead of using gold. But there's concerns about the toxicity of silver.
Now, just a last, little more provocative question. Are you afraid of causing a kind of nano syndrome, that we all going to have so many nano objects that we are not used to, nanotubes, bucking balls, and etc., that our molecules will start to change with time?
You mean that you have genetic changes, or you mean environmental?
It's everything.
Everything.
You know, and nature is not full with nano objects, right?
Right.
So we don't know what's going to happen to life
Absolutely.
if we just saturate with nano objects.
Yeah. So there are certainly environmental concerns about the release of nanoparticles into the environment. I mean, probably biomedical uses is pretty negligible, but people have suggested using nanoparticles, for example, for water remediation. So to make clean drinking water, you can use nanoparticles that have a bactericide, and certainly there you start to worry because you're putting very large amounts of nanoparticles into the environment, and we don't know what the long-term effects are. In fact, there's quite a large literature on the ethics of nanoparticles that's worth reading. So it's already reached the stage of people discussing, you know, are the social benefits of using nanotechnology likely to outweigh the social risks and the environmental risks and the health risks. So it's a serious subject for study. I mean, I don't personally have a particular opinion on it at this stage, but it should certainly be discussed. I mean, we've learned from history that the earlier you start having these discussions, at least you have a higher chance of not hitting the really big problems.
Good talk.
Thank you.
So I guess I have a basic question. Why don't quantum dots photo bleach? Are they protected from something, or is it, or what's kind of that mechanism behind that?
Because it's not a photochemical process. There's no chemical changes happening. You simply are getting essentially wavelength transformation. So the quantum dot, after it's absorbed the light and re-emits, is chemically unchanged. Whereas with organic dyes, you have the possibility of chemical change.